approach.
Thank you. I propose Steve Coles. Sorry. Okay. Yep.
I noticed that anything that goes wrong with a cell, if we want to get in and actually fix it at the detail level, we don't necessarily need to. I noticed that induction of apoptosis isn't really mentioned on there, but is that not a possibility for like many of these problems that are going wrong with cells?
That's number 10. That's number 10. But anyway, that's,
okay, sure.
That's cool.
That makes sense.
So Steve Coles, you get to do stem cell therapy to replace lost cells.
My comment actually is to defer to Bradbury on this matter, because I think pristine stem cells is the scientific area that needs our attention, because it's not well understood in any of the literature I've seen. And it's clear that we need to find surface markers that tell us whether or not the nucleus of a stem cell has been damaged. And there may be, in heat shock, tests that could be done, and other like FACS cell sorting machines that could be used in the process of identifying pristine stem cells for autologous treatment.
Yeah, given that that technology is well developed for sorting stem cells in the first place, you're doing quality control. So you could just use the standard protocols and sort through the stem cells that are circulating in the blood, subject them to some additional antibody detection methods that detect indications that cell surface proteins are accumulating that are mutant forms. Now, the only problem is that they can appear in many mutant forms. The other point I wanted to make to John is that a lot of these are relatively unfixable with a pill if the cause of them is the fact that the genome is becoming dysfunctional. Then the only solution that you really have is apoptosis and getting the stem cells in a robust way to replace the cells that you're losing. You'd ideally like a genomic program doing one-for-one replacement. I'm dying, send me assistance or send me a replacement, you know, like they do in the military, you know, kind of thing. But
point well taken. So we seem to be moving naturally to item number 10, therapies to remove inhibitory factors and dysfunctional cells. Any bidders on this one?
I can do it.
Aubrey.
Yeah, all right. So we have two ways in which we end up with too many cells of a particular type. One of them is if cells divide too often, and we're about to get to that in number 11. And the other is if cells don't die when they ought to, when cells are not necessarily proliferating, but they are supposed to respond to signals from elsewhere telling them to apoptose, and they don't actually respond to those signals. The immune system is a very important example of this. It's not just a case of thymic shrinkage causing the failure to create the right cells. It's also a case of the wrong cells hanging out for too long. And there are a number of approaches being looked at now, including in combination with thymic rejuvenation in a project that we're funding in Tucson. But there are a number of other things that could be tried. Again, this is something with relatively short-term milestones and relatively good bang for the buck, in fact, very good. It's not just the immune system, though. This is something that applies also in adipose tissue, especially visceral adipose tissue, which we've heard about from the clinicians today. That when you get too much of it, it tends to be hard to get rid of. And we've always got liposuction, but liposuction isn't so good for visceral fat as it is for subcutaneous fat. So really we want ways to stimulate the either apoptosis or conversion to heat-generating fat-burning cells of the visceral adipocytes that are excessively fat-storing. And there are other types of cell of this sort as well, cells that get into a state where they get resistant to apoptosis. There are various very good, extremely top-level gerontologists working on problems like this, but there's far too little resources in it. It's just not understood as as serious a problem as it really is. So it's definitely something that should be on our list.
Brilliant.
one additional comment.
All right, one last one on number 10.
So Aubrey's talked mainly about the cells, but I want to talk about inhibitory factors, which the Convoy Lab at Berkeley is doing very cutting-edge research on and has come out with a couple of publications in the last 6 weeks or so. These inhibit the natural ability of stem cells to repair or remodel damage, and they're also working on a blocking molecule to block the inhibitor, which is sort of a double negative, but it shows a great deal of promise. I toured their lab last month, and they said that they are indeed looking for either private companies or foundations to support their work in exchange for a piece of the intellectual property.
Cancer. Who's up for cancer? Greg.
In The Future of Aging, we have a daring chapter on cancer. It's very speculative, but it's based on many years of what I perceive to be pretty solid research. I know there's some people in the audience here who are working on immunotherapies for cancer, but the particular angle that's taken in that chapter is that one of the major defenses against cancer in the normal cancer surveillance system in the body is the granulocyte or the monocyte. And that it may just be as simple as giving people granulocyte transfusions, which has been approved by the FDA for the last 40 years or so. It's virtually side effect free. The donor is not damaged in any way and can donate very frequently. And what is alleged to happen, at least in mice, I mean, there are mice that cannot get cancer. You can inject them with anything you want, and they just don't get the cancer because their granulocytes are supergranulocytes, and they just kill the cancer. And they've set up assays showing that the same kind of phenomenology can be seen in human cancer cells in a petri dish. They've taken movies of the...
This is Genqui's work.
Yes, exactly. So not to belabor it, but it seems to me that that would be an area that might deserve some more support too. It looks like an incredibly facile way of dealing with the problem, very safe and very interesting.
Very nice. Any undiscussed forms of repairing damage that people would like to raise?
What about organ replacement? Where does that fit in? Non-biological.
Well, let's have a minute on organ replacement.
I just want to put the item on make sure it's on the agenda. There's also something else I'd like to put on the agenda. I don't know where it'll fit in, but just listening to some of the speakers, it would seem to me that one research project that could have a huge multiplier effect is to have somebody with the radical anti-aging mindset that we have in this room, who's really knowledgeable, just focus on what are the therapies being developed for other things that could be reused, repurposed, or combined in different ways for radical anti-aging.
Doesn't that define Aubrey de Grey?
Partially, I think John Ferbers also seems to be doing a lot of that. But what I'm actually talking about specifically, focusing somebody and paying somebody, raising money for that purpose to just go out there and look at, what are millions and billions of dollars being spent on something that seems unrelated but could be repurposed for radical, purposes. Okay, watchdogging. Okay, I didn't know that term.
Watchdogging. Very nice. I have you, Bill, next.
Yeah, one avenue that hasn't been yet discussed, but has been DARPA has actually been investigating, is Stephen Badylak's work at the Pittsburgh Regenerative Medicine Center, is the ability of the body to regenerate itself through studying axolotls and these types of salamanders. And that, I think, is actually where we're going to go with an awful lot of these regenerative medicine therapies.
Thank you, Kevin. The mic goes to Bill Andrews now.
I just want to add, back on mitochondria mutations, there is a company called Gensia that I was hoping would be at this conference, and they supposedly have some novel procedure now for correcting mitochondria mutations, but they're not ready to disclose it. But I think it's something we should, okay, good.
Yeah, well, you have one right there. Oh, I'm sorry. Do I? Yeah, thank you. I can say a little bit about that because I've known the people working in this area for some time. Essentially, they have a trick they've developed for getting engineered mitochondrial DNA into the body in a manner that wrapped in such a way that it actually goes through cells using protein transduction epitopes, and then also goes into mitochondria, so to actually replace the mitochondrial DNA that may or may not be mutant. So this is rather than the system I talked about yesterday of actually making the mutations harmless by putting substitute DNA into the nucleus. This is bona fide repair of the mitochondrial mutations. And they have this wonderful trick where they put a restriction enzyme, the gene for a restriction enzyme into the engineered DNA, and they engineer out the restriction sites that thing hits by synonymous substitutions, so they can make DNA that destroys the resident DNA and replaces it in this manner. It's very clever. It's something that's actually been shown to work, that part of it, the replacement part, in vitro. What's not yet been shown by any means is that this can actually be of therapeutic relevance, but I understand that Gensia have done quite well in terms of getting SBIR grants and such like. They're not really looking for money at the moment. However, that sort of work and related work around it could certainly be expanded, and I would be very much in favor of that.